Okay, fun with technology yet again. I'm not sure if it's the Mac or the projector. There's some communication issues, which I guess was also true on the exam. Communication issues, maybe, between us and you. I have the Scantrons. Of course, we're a little late now. We'll see at the end of class. Otherwise, we'll have a little bit more time on Wednesday, hopefully. You can get back all your Scantrons. All those scores are posted. There's a breakdown of the minimum grade that you would get relative to the high score. That will probably be normalized down. If you looked at the statistics, you'll notice there are at least a couple of outliers. So, and I know who you are. So we'll go from there. But first, of course, we have to have our clicker questions. If replication was dispersive instead of semi-conservative, how many bands would Michelson and Stahl have seen in their experiment after 2 generations? 1, 2, 3, 4, cannot tell from information given. Thirty seconds. Ten. 5. Okay, by the way, I did see if I could make that a little bit larger. I haven't figured that quite out yet, but I'm going to keep working on that to try and deal with this. Yes, 1, dispersive is correct. We'll choose that. I'm going to say 1 for dispersive. And the reason that I particularly chose this is because it turns out that repair, which is what we're going to be talking about for the rest of the day, also works in a very dispersive fashion. Next question. In E. coli replication, DNA polymerase I is involved in primer removal, lagging strand elongation, leading strand elongation, licensing, or A and B. Thirty seconds. Ten. 5. Okay, I, again, agree with most of you on this. Obviously, people studied for the exam, and it's good because you get some extra questions as well on that. In E. coli replication, DNA polymerase I is clearly involved in primer removal. What's going on there? Has a specific kind of activity, pretty unique to DNA polymerase I. Okay, so RNase activity not per se. It's true, but it also works on DNA just as well. So DNA and RNA are similar because of their polarity. But they also all have 3' OHs, and they're all 5' to 3'. So what DNA polymerase I does is it has a exonuclease activity that will chew in at phosphates, 5' phosphates. So it chews ahead of the polymerase that's polymerazing behind it. And so it's a 5' to 3' exonuclease. So that 5' to 3' exonuclease does chew up primers, but it's not specific to RNA. It's also will work on DNA. Yeah?
What's the difference between an endonuclease and an exonuclease again?
Yes, so we're going to get more to that as well a little bit later on. But endo is in the middle, exo is at the end. And so endo is getting the middle of your DNA, and exo is getting the end of your DNA. And unfortunately, those of you who've seen me on campus in previous years, I used to have this wonderful DNA model that I carried around, but it seems to have disappeared. But yeah, so it's the ends of DNA. And it turns out ends of DNA, which is also really important for repair, are very unstable. And there are tons of exonucleases around in the cell. As soon as you have a free end, that's going to get chewed up. And so it's the DNA polymerase I that does that, and lots of other exonucleases. At the same time, it's a DNA polymerase. So it will extend also in the 5' to 3' direction, always on 3' OHs, but that's always going to be on the lagging strand, because on the leading strand, you have DNA polymerase III. Once it gets on, it stays on and keeps going. And so for that reason, E is correct. Leading strand is only DNA polymerase III. And licensing, it has absolutely nothing to do with. Third clear question for today. DNA polymerases add, not well proofread here, deoxyribonucleotides to 2' OH groups, to 3' OH groups, 5' phosphate groups, A and C, or B and C. Thirty seconds. Ten. 5. Stop. Yes, we all agree. That's good. It's really critical that DNA polymerases can only extend from already extant primers. And so that's the really critical aspect of this. If we can actually select the answers now. Is it going to let me? No. Oh, actually, I have to stop first. That might help. Good. So today, we're going to talk about DNA repair. And the DNA repair aspects are such that, DNA, as it turns out, is actually a really pretty crummy molecule to use for your genetic information. And it tells you that evolution is really a case of good enough, as opposed to necessarily the optimal way of taking care of your genetic material, or all the genetic material that we know of, with the exception of viruses. Again, always going to be exceptions. Talk a little bit about the types of damage that can happen, particularly the really common types of damage that happen to DNA. And it turns out just the fact that we live in an aqueous environment and our internal temperature is about 37 degrees Celsius is really dramatically damaging to DNA. So those are sort of the kinds of damage. How do you deal with that damage? Base excision repair. This is really going in and very specifically, so think of the scalpel or the laser surgery for dealing with DNA repair, or nucleotide excision repair, which is much more of the chemotherapy sledgehammer approach to dealing with DNA repair. And then a very specific problem, this gets back to the comment about exonucleases, and that is that with exonucleases, you've got ends, and ends are really unstable. Whenever you break both strands of DNA, it's a really huge problem because you don't have that backup copy. Normally you've got that backup copy when you've got double-stranded DNA. And double-stranded breaks, of course, you don't have that backup copy anymore. Two different ways of dealing with that, either non-homologous end joining, which is just grabbing ends and putting them back together, which, not surprisingly, is a pretty dangerous way to go, or homologous recombination, which seems to be the way that most double-stranded DNA repair takes place. So why is DNA a bad genetic molecule? Because everything in DNA can basically be chemically modified, relatively easily, in fact. And the chemistry of the DNA, of course, is critical for that information. It's all in the base pairs, and literally the arrangement of your base pairs in terms of that genetic information. Once you've changed that genetic information chemically, that can be inherited. So a very dangerous process. When you talk about any one of these particular changes, and when people talk about mutations, basically a mutation is a change in the genetic material. And that change in the genetic material, of course, can be inherited, but that change can also mess up replication, so you can't make more copies. It can mess up transcription, so you can't be making the RNA. Probably mutations are involved in cancer. Certainly you have lots of mutations in cancer. Whether you need mutations for cancer is a little more questionable, but I think the evidence is really preponderance for, yes, mutations are really important for cancer, and they may, this is a lot less certain, be important for aging. The saving grace, of course, of DNA is that most of the time it's double-stranded, and so you've got a redundant copy all the time. And so that's true as long as the two strands are still together. The other thing, of course, and this is probably why this crummy genetic molecule is actually used, or has been maintained through evolution, 3 to 4 billion years thereof, at least on this planet, that allows evolution. And so some of the changes that happen turn out to be beneficial, and there's selection for that particular beneficial changes. We know there are lots of human diseases, again, above and beyond the cancer. Yeah, sure. If I do that, I'm afraid it'll stop. So I was trying that earlier. I don't know if you were here right in the beginning. So yeah, sorry about that. I would if I could, but I'm worried that it's going to break. Yeah, so human diseases of DNA repair. So again, above and beyond the aging and cancer, there are a number of inherited diseases that we know are really problematic. And these, as it turns out, are diseases where patients, this is human patients now, have problems in a number of different genes, be they mismatch repair, nucleotide excision repair, translesion synthesis, et cetera, et cetera, et cetera. So lots and lots of congenital human diseases have to do with DNA repair. And so also gives you a good indication that DNA repair is a rather important process. What are the problems? Well, basically we live in an environment where DNA is not particularly stable. Fortunately, we don't have to deal with ionizing radiation, at least not most of the time. Turns out that ionizing radiation that probably most of us will ever have to deal with is actually being used as a therapeutic method. It's trying to break the DNA. So radiation medicine, for instance, this is all about making double-stranded breaks or breaking the backbone of the DNA. Chemical-based modification, this happens all the time, and just a back-of-the-envelope calculation, particularly for cytosine, the amine comes off of that cytosine at about 100 per cell per day. And you think of the trillion number of cells that you have in your body, this is a very, very common. Even more common is depurination. Those large bases, the adenines and the guanines, have a nasty tendency to pop off of the backbone, and they do this just because we happen to live at a 37 degrees approximately internal temperature, so 5 to 10,000 per cell per day. This deamination takes place. And so it's a really common process, and of course if these are common processes, these are things that the cells have evolved mechanisms to deal with. So we'll talk about that. UV light is another big issue. That's one really good reason to wear sunscreen and live in the Pacific Northwest, is we don't have to deal with too much UV irradiation, but it causes some really nasty problems, particularly pyrimidine dimers, which also then need to be repaired. So the basic problem is we like it warm, we like oxygen, and we like light, which are all bad for DNA. So let's look at these particular kinds of mutations that take place. Would it be better if I tried to lower the lights for this, or does it really matter? Let's see. Can somebody over there maybe hit one of the light switches? The bottom one would be best on the right-hand side, if you would. Oh, and that panel. Is that better, worse? Better? Okay, good. Thank you. So there are really two kinds of mutations that happen. This is now a mutation which is being transmitted potentially on replication. You either have the easy mutations or the hard mutations. Transitions are going from purine to purine or pyrimidine to pyrimidine, so small to small or big to big. And the way that most people look at these, Y is for pyrimidine, so this could be cytosine or thymidine, or the purine, which could be what? Adenine or guanine. If ever you have more drastic changes, so you go from a small to a large, or a large to a small, these are going to cause much more changes in the structure of the DNA. Because if you think about it, you've got two purines that are trying to base pair with each other. There's not enough space between the two strands, and so it's going to be pushing it away. And so these transversions are actually easier to detect as terms of the molecular machinery than any of these transitions. And it turns out that transitions are much more common, or commonly not repaired, in DNA repair mechanisms. So let's look at what happens here. Now, classic case of problem with replication. You start out with your parent molecule here. There's a replication error, and almost always these replication errors are going to be small to small or big to big, because they still fit in the structure, the double strands, a lot better than the opposite, which could be taking place. Once you've had an error, if that doesn't get repaired, then of course in the next generation you'll be changing that opposite strand. Now you have good base pairs, everything is happy. So there's this window right here where you have to repair the DNA, or again, in some cases it won't be repaired, which can serve as the raw material for natural selection and evolution thereafter. Another thing that happens, and this is something we've just discovered much more recently, is that quite often DNA polymerases will do something they call slip, and that's when you have nucleotides that are absolutely identical to each other, and particularly common, these are nucleotides that are repeating one after another. So in this case, GT, 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 GT. Of course, in the opposite strand, that's going to be AC, AC, AC. That the polymerase just slips and will slip back and make extra copies of these particular, usually very short sequences. And we see this as happens in all kinds of different organisms, be it E. coli, humans, etc. So these repeat units are very often added to DNA. And in fact, they turn out to be really useful. Have anybody heard about microsatellites? Anybody work with microsatellites? But these are really nice genetic markers that you can use to follow populations with. And it turns out that they're all having to do with these additional repeats, which are added by the DNA polymerases. So let's look at the DNA polymerases and how they actually do this. First, most of the time, DNA polymerases do a pretty darn good job. They don't actually put in incorrect nucleotides. But what's interesting for the DNA polymerases is they're not actually looking to see whether an A has paired with a T, or they've got an A on their template, they know they have to bring in a T. And this is a bit unlike the cases happening with the ribosomes. They just look to see if the geometry is correct that you have in your DNA molecule. So if you have something that forms a nice base pair, so it'll be an AT or GC or TA or CG, then that's going to have a really consistent structure. And so really what the DNA polymerases are doing is they're checking to make sure that the structure is correct. They could care less about the sequence. They're just making sure that the structure is correct. And it turns out that it's those base pairs which actually work there. So you're looking at base pairs, and all this happens through stacking. Base pairs, of course, have these pretty large ring structures. They're pretty planar. All that you do is you take a planar amino acid and pop it down on top of that. The particular one here is tyrosine. This is true in pretty much all DNA polymerases. Put in the new nucleotide, check. Folding down the tyrosine, if it folds down properly, you have hydrolysis that takes place. If it doesn't, you don't. So it's a really pretty straightforward process. And again, if you don't have the right base pair, you're not going to have this flapping down. You're not going to have catalysis that takes place. So that's one thing that happens. And it turns out that's actually pretty darn good. 1 in about 10,000 nucleotides will slip by this. If it turns out that catalysis does take place and you do add the nucleotide, then you have another kind of exonuclease activity. And this is the backing up exonuclease activity. So we already talked about the case for DNA polymerase 1, which is a 5' to 3' exonuclease activity, chewing ahead in the same direction you're polymerizing. But the proofreading activity is 3' to 5', so it's going back. And it's taking out the nucleotide, which turns out is the one which is the incorrect nucleotide which gets put in. And again, this is all detected by structure. It's, makes a nice base pair, doesn't make a nice base pair. If it doesn't make a nice base pair, what happens is that pairing, you have just shown here on the top, pops off this base which is not properly paired, chews off that phosphodiester linkage, and then continues to extend. And so this is the so-called proofreading activity. Most DNA polymerases, not all of them, but most DNA polymerases have this proofreading activity. The proofreading activity takes place through a particular domain of the DNA polymerase. And so we talked about domains before. These are separate parts of the protein, fold up into their own structures, and very often that structure is going to give you a function, and this particular function is a proofreading exonuclease activity. So a backing up exonuclease activity. So that's a specific domain that takes place. Again, it's the configuration which is detected, and then once you've backed up, all that you've done is you've cleaved the phosphodiester backbone. You've cleaved it at exactly the position between the phosphate and the OH. That gives you a 3' OH. You've got a 3' OH. You've got a template. The DNA polymerase is happy. It can keep going on that. And that is probably why we only have polymerases that go from 5' to 3'. And the reason for that is if you were to be going in the opposite direction, you're going to be adding on phosphates all the time. Then if you were to cleave off that last nucleotide that you made, you wouldn't be able to extend it anymore. Because if you think about it, these are triphosphates, all the triphosphates that you come in with. So that triphosphate, NTP, somebody asked me about the N. The N can be any of the 4 nucleotides. There's an alpha, beta, and a gamma phosphate that hangs off of it. The polymerization reaction is cutting off the beta and gamma phosphates. And those are then released, which is fine as long as it's attaching to an OH. But if you were trying to attach an OH to a phosphate, and you just cleaved off that phosphate at the end, you're not going to be able to add back to it. So basically, because of the chemistry, you have to have 5' to 3' polymerization. And if you didn't, you had 3' to 5' polymerization, you wouldn't be able to repair it, at least not with this kind of proofreading mechanism. And it turns out that proofreading gives you about a 100-fold increase in your fidelity. So as I said before, this is your polymerization, just checking the structure, about 1 in 10,000 you mess up with. If you add proofreading, now it's 1 in, so that's 10,000, 100,000, in a million or 10 million. And then we've also got strand-directed mismatch repair that we'll talk about in just a second. That's if proofreading doesn't work and you still put in the wrong nucleotide. That then gets you to about a 1 in a billion. And sorry if it's hard to see at the bottom here, but it's about 1 in 10 to the 9 base pairs gets incorrectly added per replication cycle. And if you think about the human genome, it's how many billion base pairs? About 3 for the haploid genome, 6 if you think about the diploid genome. So that means every replication you're putting in 6 mistakes. Which is, not that many, but certainly enough to give you, again, the raw materials for evolution to take place. So this is getting back to your question about exo and endonucleases. Here's just a DNA where you have a break in the phosphodiester backbone. This now provides a site for exonuclease activity because you can cleave away at this end. You can either have a 5' to 3' exonuclease activity, which will go in this direction, or 3' to 5', which will go in that direction. But you also have an endonuclease site, which is right here. It's cutting in the middle of the DNA. So again, DNA polymerase I has this exonuclease activity and for proofreading, but also an exonuclease activity for getting rid of primers. And that's the big difference with DNA polymerase I relative to all of the other polymerases that we've talked about in this class so far. So that's what happens with just normal replication. So 1 in a billion or so, you're going to get changes. But, of course, as I mentioned before, we've got this nasty environment that we happen to like to live in: water, oxygen, and light. Particularly in terms of chemical changes, the main issue is water and just hydrolysis. We're 80% water. It's all about the water in terms of any of our cells. So all of the blue arrows down here at the bottom here represent places where you get hydrolysis. And the important ones here are right here, and this is the glycosidic bond between your deoxyribose and the base. Here, for purines, the size of the arrow here corresponds to how often it happens, and depurination over here for your adenine bases. So, cuts that, you lose the adenine residue. The other important one right here in terms of hydrolysis is deamination of cytosine. So you see this blue arrow up here, lose the amine off of your cytosine. There are a number of other things that happen, but we're going to concentrate on these guys. Oxidative damage, the most common oxidative damage is in guanines and adenines. And then we'll talk much more about methylation later on. Methylation is a DNA damage, but also can serve as a signal. And we'll talk about, again, the signal much later on in the class here. Most common, again, these are the depurinations, so loss of those purine groups, or deaminations, particularly deamination of cytosine. So that's, again, this is shown here. Depurination, have a guanine, hydrolysis. Now you have your ribose that doesn't have a base on it anymore. The other case, deamination of cytosine. Here, lose this amino group, and what happens is your cytosine actually gets changed into uracil. And this is probably why uracil is used in RNA, but not in DNA. Because the DNA can recognize that you now have uracil in a DNA strand, and now the molecular mechanisms know, hey, deamination must have just happened. And so if deamination has happened, then we know that this is wrong, and we want to take it out. And so that's the process here. So most common DNA damage, again, depurination and deamination. Other ones that happen as well. There are lots of different ways to repair these kinds of DNAs, but we're mostly going to spend our time talking about these two here in the middle, B and C. Direct repair, this is probably actually the most efficient. It takes one particular change, and from that one particular change, it changes it back. So happen to know exactly what the problem is, how it happened, you can put it back in. Usually, it's not that easy. On the other hand, excision repair is, again, this is the sledgehammer approach that I talked about, which is where you chop out a piece of DNA and then put back in new, depending on having that opposite strand that you have there. Mismatch repair, okay, this figure has it as being different than excision repair. It's actually pretty much exactly the same. Again, you cut out a little piece and then you put it back together. Non-homologous end joining, this is where you have those double-stranded breaks. And the problem here, of course, as I mentioned before, you don't have that opposite strand. You don't have your redundant strand to deal with. And so putting the two strands back together is a dangerous thing to do, but you still have to do it in some cases. It's much better if you can use some of these other ones, particularly recombinational repair down here at the bottom. So let's look at the two sort of major ways. Oh, sure. Yeah?
I didn't know if you were going to go into these more, but for mismatch repair and excision repair, do they both cut like on both sides of the damaged nucleotide, or?
Give me about 2 minutes.
Okay,
Yeah, so we're just about to get there. So yeah, two different ways of looking at that. And again, so there's the surgical approach and there's a sledgehammer approach. Surgical approach is base excision repair. So it's, just like it says, a single base, and literally a single nucleotide. And then nucleotide excision repair, which is chopping out a big piece on both sides of where you have the problem. So these processes are really quite different from each other, particularly at the beginning. The first one is detecting that you have a problem, and usually this is going to happen for those problems that are happening really frequently. So deamination, depurination. So these things happen very frequently. Cells have evolved systems to deal with these particular changes. So you recognize the problem. In this case, it's a uracil, which has come from deamination of cytosine. There's a very specific protein that looks through DNA, finds uracils, and chops them out. Once they've been chopped out, now you have a strand which is missing a base in the middle. This actually looks a lot like depurination. So once you're missing this base, this is another very common kind of DNA damage. Then there are specific endonucleases, so it's cutting in the middle of your DNA, which will come in and cut out one of those backbone nucleotides. So it's got rid of the phosphate, it's got rid of the ribose, and then DNA polymerase will put in a base. You need a DNA ligase to hook those things together, and now we have happy DNA again. Now, this only works because we have a very specific enzyme at the top here that recognizes this one specific change. So really common changes is great. But if you've got things that are very different kinds of changes, or things that don't happen as often, you haven't developed, again, evolutionarily, ways to deal with that. So if you have a bigger problem, then that's where you get the nucleotide excision repair, which comes in. It recognizes that there is a problem, but not that there's a specific problem. Once it sees that you have a problem, then you have, again, endonucleases that cut on either side of that problem. And then you've got templates, you've got 3' OHs. Those are ready to go as long as this DNA has been gotten rid of. It's a DNA helicase, just pulls that piece off. And then DNA polymerase and DNA ligase repair your DNA down here. Let's look at each of these again in slightly more detail. Here's deamination. Deamination, again, happens. We happen to live in an aqueous environment. Here is a uracil residue in your DNA. This uracil residue in the DNA is bound to and cut off by the uracil DNA glycosylase. What to me is pretty amazing is that these are enzymes that have to be checking the DNA all the time. Every single base in your DNA is being checked all the time by these glycosylases, which is pretty amazing. Again, think about it, 6 billion bases in each of every given cell. They're being checked all the time. The other thing, this is a reminder that we always see the wonderful, double-stranded DNA models, but it turns out that all of these models are splendid and worthwhile, but they're very static, and DNA is a very dynamic molecule. It flops bases in and out all the time. They're coming apart, coming back together, and so that's why you can actually have these DNA glycosylases that will recognize these particular structures. And again, these are pretty classic deaminations that take place. The one here in the middle, this is the cytosine deamination. So let's get rid of this here. Cytosine deamination, giving you uracil, but also other deaminations take place. Adenine goes to hypoxanthine, guanine goes to xanthine. Don't expect you to remember these names, but you get other kinds of deamination that take place. None of these are natural DNA bases. On the other hand, 5-methylcytosine, which, as we'll see later, is a very common modification, actually is important for DNA methylation, not just in bacteria to show that you've gotten replication to take place, but also in eukaryotic cells like us. So a lot of our cytosines are methylated. Now, why is this a problem? It's because if you now deaminate 5-methylcytosine, you end up with thymine. And thymine is normal. If you were to go through your 6 billion bases of DNA and cut out all the thymines, you'd have all kinds of problems. So it turns out that almost all of the spontaneous mutations that happen in human and, in fact, most eukaryotic DNAs are these changes. So 5-methylcytosine gets deaminated, you get thymine. When that doesn't get repaired, you replicate, and now you've gone from a C to a T. So again, it's a small, these are pyrimidines, so nice transition that takes place. The opposite strand, of course, has now gone from G to A in terms of the base pairing that takes place here. So most common mutations that happen, again, having to do with 5-methylcytosine, which is there. Again, this is basically the same process. Here's another picture of one of those bent DNA molecules binding to a specific DNA glycosylase. Turns out it's actually not the uracil DNA glycosylase, but it's an oxidative form of G, which also gets cleaved. But again, you cleave off one nucleotide, you end up with a ribose without the base. That gets chopped out, and then your DNA polymerase comes in and takes care of it. This is the figure from our textbook. I like the other ones better, but again, basically exactly the same thing that takes place there. So the next thing I wanted to talk about are these bigger problems. And these are the ones which are, again, are not so specific, they're not quite as common, but still, clearly are going to be issues. And probably the most common of these, again, much less so than deamination and depurination, is thymidine dimer formation. And this is caused mostly by UV irradiation. Happens any times that you happen to have two T's or a C and a T or a C and a C that are right next to each other in one strand of the DNA that happens to pick up some UV irradiation. What happens is they make these dimers, covalent bonds between the two pyrimidines that are stacked on top of each other, and that basically takes the DNA, which is normally twisting around itself, and then locks these two bases standing right on top of each other. Not surprisingly, what this does is it changes the DNA structure. So if you've got normal B-form DNA here in green, as soon as you make a pyrimidine dimer, that kinks the DNA. And it's that kink which gets detected by the DNA repair machinery. Not surprisingly, because this mostly happens due to ultraviolet irradiation. Most of the proteins are known as UVR for UV radiation, because these were all the geneticists. What did they do? They found mutants that were sensitive to ultraviolet radiation and then figured out what they did. These are the UVR A, B, and C proteins. This is from E. coli. Turns out it's much better understood in E. coli than in human cells, but the mechanism is practically identical for how these things work. You have UVRA that binds to that damaged DNA, and it's not so much the damage itself, it's that structure. It's the weird structure. there's a weird structure in your DNA, then UVRA will bind to it. UVRB basically seems to come along for the ride with UVRA. UVRC is that critical endonuclease that makes the cuts on either side of this problem and then allows the next steps to take place. So I just said this in words. We'll look at it in pictures right now. Here is the problem in your DNA. It's a structural distortion that you have in your double helix. UVRA and B bind to this. There's ATPase activity. That ATPase activity basically seems to be involved in rearranging the structure of UVRA and UVRB, just to say, hey, we found something which is wrong. Then you have the association of UVRC. This is that endonuclease that cuts in the DNA on either side of where you have the problem. Then there's the UVRD helicase, and then DNA polymerase, got a 3' OH, it's got a template, will replicate its way through this. Human cells do this in an extremely similar way. Here again, we have a thymidine dimer. These are the proteins which are involved in taking care of that. Many of these proteins, excuse me, were discovered because of patients who had problems dealing with DNA base pairs. So the particular ones here are xeroderma pigmentosum proteins, also known as XP. Sorry, maybe a little hard to see here, but XPC, xeroderma pigmentosum patients have a huge rate of skin cancer. And that's because UV irradiation causes these problems. They don't get repaired properly. So, not surprisingly, a lot of these proteins are mutated in those patients. So, this xeroderma pigmentosum protein binds in a manner very similar to UVRA, UVRB. That then leads to cleavage on either side of that mutation by endonucleases. This is the XPG protein. And then you have helicases, XPF, which will then get rid of that particular piece of DNA. It gets repaired. There's one particular thing to mention in here. The book makes a pretty big deal of it, and there's also really a big surprise when people first found it. They had these xeroderma pigmentosum patients, and they were looking at where all the mutations were in their particular proteins. And it turns out they didn't just have problems in repairing the DNA, they also had problems in transcription. And it turns out that there's one particular protein that we'll spend a lot more time talking about later on, which is involved both in transcriptional elongation, but also in helicase activity for getting that strand off of the DNA, the one that's got the problems in it. So this is the TF2H protein that, again, we'll spend a lot more time talking about later on. So nucleotide excision repair, base excision repair. There's a sort of modification that I like to think of called excuse me, mismatch repair. But if you think about a mismatch, particularly if it's a purine-purine or pyrimidine-pyrimidine mismatch, that's going to cause a problem in the structure of your DNA because your base pair is not proper. So the base pair is not proper. Not surprisingly, that's going to cause the machinery very similar to the nucleotide excision repair, which is going to come in, cut on either side, and take everything out. The difference here really has to do with the individual proteins, but also how do you know which piece of DNA to take away? Because if you have a mismatch that you've put in, and usually this is happening through the activity of your DNA polymerase, you need to know what's the wrong one and what's the right one. Now, how do you get rid of one versus another? In the case of E. coli, it's pretty easy because, if we remember from licensing, methylation. The old strands are the ones that are methylated. The new strands are not methylated. If you have a mismatch, which one do you think is the one that you want to take out? It's the one on the non-methylated DNA strand. So that's pretty straightforward. The proteins that do this are called the mute proteins for mutations or hypermutations. You make a mutation in these particular proteins. You end up with lots more mutation because you're not taking care of these mismatches that get added. But the process is basically identical to nucleotide excision repair. You have binding to the structure of DNA. You have endonuclease activity on either side, and then this gets helicase activity degraded and resynthesized by the DNA polymerases. The human mismatch repair is basically identical, and one of the reasons I also mentioned the E. coli, A, again, it's a wonderful model system, but B, these guys were found because they were homologs to the mut-S protein. So MSH is homolog to mut-S. So it's a protein that was found in human cells. They looked at the gene sequence and saw it was similar to what had been really well characterized in E. coli in terms of mismatch repair. And so people assumed that these would be similar for mismatch repair, and it turns out that, yes, it is almost exactly the same kind of function. These guys, again, bind to DNA, which has the wrong kind of structure, then endonuclease activity, helicase, and get these resynthesized. One of the big questions is, what strand do you want to deal with? Because in eukaryotic systems, you don't have differential methylation between old strands and new strands. Licensing is taken care of by phosphorylation of those origin recognition complexes. So it's got to be something different here. Well, as I mentioned before, probably one of the most common mutations that happens in human DNA is that you have a 5-methylcytosine that gets deaminated, and that goes to a T. So these are the most common ones that take place. Yes, I know I have office hours right after class. So you have a transition which takes place, TG base pair. That TG base pair is probably because you used to have a C there that was methylated, that got deaminated. So we want to get rid of this guy. So you can get rid of it by mismatch repair, but it turns out that there's also in human cells a specific enzyme that actually recognizes TG base pairs because it's so common that you have methyl C that gets gotten rid of, deaminated, I should say. And so it actually literally cuts out T's in TG base pairs, and then this works much more like base excision repair. And base excision repair, again, DNA glycosylase, AP endonuclease, gives you a single base gap that then gets repaired. So in terms of base pair repair, so say the nucleotide excision repair, base excision repair, mismatch repair, there's only one more thing that I wanted to talk about. And this is kind of getting back to that issue about TF2H, again, the transcription factor that's involved in dealing with nucleotide excision repair in eukaryotic cells. Well, it turns out that if you look at just where mutations take place, and these are the ones that are maintained generation to generation, they're usually in parts of the genome that are not very active. They're in parts of the genome that are not being transcribed very often, et cetera, which means, of course, that areas of the genome that are being transcribed quite often actually have better DNA repair. There's more DNA repair that happens in these regions. So the question is sort of why that happens. Well, in the case of eukaryotes, if you've got TF2H, which is part of your transcription complex, you're transcribing a particular piece of DNA, that protein is already there. And so that's probably another reason why we have the helicase activity of a transcription factor, which is also involved in DNA repair. Turns out that the same thing is true for E. coli. In E. coli, you have association of these nucleotide excision repair proteins with your RNA polymerase while it's transcribing along DNA. And what seems to happen is once it runs into a problem, and again, this is a problem with the structure of your DNA, not the base pairing per se, the polymerase gets stopped, and then you have the nucleotide excision repair proteins that come in. And this is a nice review. This is to give you an idea that this is really quite active area of research. This is from Nature a couple of weeks ago. Had a nice review about this. Here's the RNA polymerase, which is transcribing its way along the DNA. Comes to a problem, which is where this star is. You have UVRD, and then another protein we'll talk about later called NACE. This then repairs this particular problem in the DNA, and you can go through, take care of this problem. On the other hand, it turns out this a newly discovered pathway, which we didn't know about at all until last year, which is yet another way that the RNA polymerase can deal with these particular changes. And it's probably also true in eukaryotic cells, not just in bacteria. So again, what we tell you now in a molecular biology course, by the time you look at it again, if you ever do, in a couple of years, there'll probably be more mechanisms, more different ways to skin the cat, as it were. So now, switch gears really kind of completely. Let's start talking about double-stranded break repair. How do you get double-stranded breaks? Well, again, it's mostly due to ionizing radiation. And again, this is something that hopefully most of us don't have to deal with most of the time, but it can be extremely useful in terms of therapy, making double-stranded breaks. And it turns out that not just ionizing radiation, but also quite a few chemotherapy drugs make double-stranded breaks. And the reason for that is these are really nasty kinds of mutations that take place. The idea is that that causes those cells that have the double-stranded breaks in them to actually be degraded, and sometimes degrade themselves. How does this actually happen? Either the ionizing radiation actually hits the DNA directly. Turns out this is probably a little less likely or less frequent than the so-called indirect damage, and that's basically that you ionize water. Water's got this wonderful oxygen molecule that's associated with it, so it gets hit by ionizing radiation, turns into superoxide, can turn into hydrogen peroxide. All of these are highly active molecules which can act on the phosphodiester backbone. So ionizing radiation can cause this. The reason that ionizing radiation is lethal, be careful when you're working in power plants, is because of these double-stranded breaks. What happens when you have double-stranded breaks? You can delete DNA, duplicate your DNA, invert your DNA, translocate your DNA, all kinds of problems, because once these two ends are not attached to each other, putting them back together is a pretty random process. So it's a real problem that you have to deal with. Two different ways of dealing with that. One is non-homologous end joining, and that is just like what it says. Get two ends that may or may not be similar to each other, and puts them together. Rather dangerous process, but if you have to put the ends back together, because you've got to be able to replicate, etc., you're going to do it. Homologous recombination is a much better way to go, because what you do for homologous recombination, you find a piece of DNA which is similar to the piece that you need to repair, and basically use that as a backup copy. And so you don't have your double-stranded DNA because those strands have been separated from each other, but certainly in all diploid organisms, you've got two copies. They may be different alleles relative to each other, but you've got two copies. So you can use that other copy of the DNA as a backup copy for looking at it. Overview, how this works. Two different ways of solving the same problem. Non-homologous end joining. Basically, you have this double-stranded break. What happens in these double-stranded breaks, it's very rare that you have a double-stranded break where you have a break exactly two strands opposite each other. It's going to be offset a little bit, usually. And it turns out that the mechanism of repairing this problem is such that you end up chewing in at the ends. I mentioned before there's lots and lots of exonucleases in the cell. As soon as you break one of those ends, it gets chewed in a little bit. So what happens is you chew in, and these are, in fact, some specific nucleases, exonucleases that do this, that chew in the ends a little bit, and then we'll put those ends back together. And just the fact that you have to chew in the ends to get nice flat ends that you can hook back together, you're automatically losing some of the genetic material that way. And that's just the process of non-homologous end joining. You always have to cleave in the ends a little bit before putting them back together. On the other hand, homologous recombination, you lose DNA from where you have made that particular double-stranded break. And actually, it's a very similar mechanism as you have over here. But instead of banging those two ends back together, now you use the information from your homologous copy to repair it. So we'll spend a little while talking about those, particularly non-homologous end joining, and then homologous recombination we'll talk about more on Wednesday. Non-homologous end joining, again, you start out with a double-stranded break in your DNA. These proteins called Ku bind to these ends. Then there's a DNA protein kinase. What does it do? It says double-stranded DNA, phosphorylates things. What does it phosphorylate? Particularly phosphorylates the enzymes that are involved in putting these ends back together. In order to put them together, you have to have exonucleases that cut back a little bit. Then these things get put back together. This process, again, leads to small deletions, usually less than 10 bases, which then you get put back together. Normally, people thought that, hey, we've got so much junk DNA in our genome, it doesn't make a difference if we chop out a couple of bases and then put them back together. But as we now know that most of our genome is actually not junk DNA, so this may be a much more dangerous process than we thought of. But it turns out that evolution, not being anthropogenic here, but has actually used this process to generate antibody diversity. Turns out that Ku and DNA protein kinase are used for what's called somatic hyperrecombination. And basically, wanted to learn more about it, take immunology next term. But the process here is really about generating huge amounts of diversity in the antibody-antigen interaction. So making lots and lots of different antibodies. The way you do that is by chopping out a couple of nucleotides all the time, each time when you put two pieces of DNA together. So yes, it's a dangerous process if you're talking about it in most cells, but if you're thinking about it in terms of generating antibody diversity, it's actually a really good mechanism for dealing with that. So I think at that point, before we get into homologous recombination, we'll stop.